This is video 2, amino acid graphs. Last time we saw that each of the 20 amino acids has a distinct residue structure. The residue structures are shown on this page. Consider the residue structure of the amino acid shown labeled B. We can quantify the structure using graph invariance. For example, we can find the domination number or the longest path. However, we will not be able to distinguish between these two structures. Notice that they both have the same residue structure. Consequently, they will have the same graph invariance, but they are not the same amino acid. We see that, on the periphery, one has 2 oxygen atoms, while the other has an oxygen atom and a nitrogen atom. In order to use graph theory, we need to incorporate this difference into our graph measure. In order to use graph theory, we need to incorporate this difference into our graph measure. Consider the degree of a vertex. We can think of it as the sum of the weights of its neighbors, instead of the number of neighbors. Now, if the vertices have distinct weights, the sum of the weights of its neighbors incorporates this fact. Now the sum of the weights of the neighbors is 7. Returning to the amino acid example, we can weight the vertices by their corresponding atomic mass. Then, using the sum of the neighbors as a weighted degree measure, we obtain a different value for each. This shows us how vertex weighting can be a valuable tool. Vertex weights are not highly studied in graph theory. However, chemists use graphs to study small chemical molecules, and they sometimes use vertex weight. Chemical Graph Theory by Nedetrajstic. Introduction to Chemical Graph Theory by Daniel Bonchoff are 2 of the best known books in chemical graph theory. Topological indices together with quantities derived from chemical properties are called molecular descriptors. There are over 3,000 molecular descriptors in the Handbook of Molecular Descriptors. The tool of QSAR, which stands for Quantitative Structure Activity Relation, model the process by which chemical structure is quantitatively correlated with a well-defined process, such as biological activity or chemical reactivity. The valid band index is given by a formula, where SI is the sum of the distances of the vertex to the other vertices, Q is the number of edges, and mg is the cyclomatic number, which is the minimal number of edges whose removal results in an acyclic graph. For example, we used this graph earlier when studying distances in graphs. This measure can be calculated by summing the rows of the distance matrix, similar to the Wiener index. This measure is one of the more widely used chemical topological indices. The 2 types of graph theory we are discussing are mathematical combinatorial graph theory and chemical graph theory. Combinatorial graph theory, developed independent of chemical graph theory. Typically, combinatorial measures have a max-minimum type definitions, whereas topological indices are defined by a computable formula. There is a stark contrast. Chemical graphs versus combinatorial graphs. Computable versus not computable. Small graphs versus arbitrarily large graphs. The question is, which one is best to model protein structures? We have seen in this class that networks are currently being used to model protein structures. Measures such as average path length, clustering coefficient, and centrality measures can capture some of the global properties of protein molecules. As you know, this is built on the earlier work by Watts and Strigatz and the small world phenomena. There are pros and cons for each of these methods. For example, chemical graphs measures are well-developed concepts with computable values. However, Best results are for small graphs. Combinatorial graphs measures are also a well-developed resource, but in general the measures are intractable, and care and extra work must be undertaken when finding them. We have found that we get the best results when we use all of these combined. How can we best model protein structures in order to inform us of the characteristics of their respective functions? Given the variety of protein structures, it is not surprising that there is not the one-size-fits-all method.